Good afternoon, everybody. The light is so beautiful on the campus. I'm sorry to tear you away from it, but I promise we'll have fireworks here for you with a brilliant lecture and an amazing event here at Mills. So I want to thank you all for joining us for the Russell Women in Science Lecture. This is a lecture series that has been running since 2012. I don't have the pleasure of introducing our speaker to you, but I do want to thank her for being here. Thank you, Dr. Cori Bargmann, for joining us today. Dr. Bargmann has been on our campus talking with Mills' greatest asset, our students and our faculty throughout the day, and also experiencing our campus, which is in full bloom in spring, is helping us put on a great show too. I do have the great good fortune to welcome Chris Russell, who's here, Mills, a proud Mills alumna, a trustee for many years of the college, an accomplished science writer and distinguished teacher herself, who will have the pleasure of introducing Dr. Bargmann. But I do have to say a little bit more about Chris before we get going, and thank some other folks. So I want to thank our advancement staff that helped set up this event, and Dr. Lynn Delker, who was also critical to this. Thank you, Lynn. I want to thank each of you who have taken time out from your busy day to join us here. So the Russell Lecture on Women in Science has actually played a big role at Mills over the years. Some of you have had a chance to experience this before, and some of you are here for the first time, and we're glad for all of you. I'm going to pause for a moment to talk about something Chris asked me to mention too, that involves my own experience with women's leadership in science. So last year, I was part of a National Academies of Science, Engineering, and Medicine study committee that was chartered by the Committee on the Status of Women to look into the persistence of sexual harassment in academic STEM fields. That report was issued last year. I had the good fortune to teach a course with some Mills students where we read that report carefully, and I've had the chance to talk about it around the country. If you haven't had a chance to read it, I recommend it, but the main conclusions are, if you didn't know sexual harassment was a big problem, it is. If you didn't think it had significant consequences, it does. If you thought what we have been doing is working, you're wrong. And we actually do know what will work to actually address this problem, and the report lays out some recommendations. Today, we issued a press release for the Action Collaborative on Fighting Sexual Harassment. That is, the National Academies put this out. Mills is a charter member of that group. We were one of the founding members. Now there are 40 colleges and universities that are joining an Action Collaborative to undertake a structural, data-based, long-term approach to solving a problem that we have not yet addressed, that will support gender and racial equity in the sciences in ways that engineering, medicine, and science, as fields that have attracted increasing numbers of women but have not solved this problem, haven't managed to do. So I'm excited that we happen to have a brilliant leader and scientist here on our campus who has surmounted some of those challenges that face women in these fields, as well as a distinguished alumna to introduce her. So let me say a few more words about Chris. Her visionary commitment to women's leadership in STEM fields is the reason we have this particular program at Mills. She's an award-winning freelance journalist. She's written about science, health, and the environment for more than 4 decades. She was a biology major here at Mills. She's a former member of the Mills College Board of Trustees. She's currently an adjunct lecturer in public policy and a senior fellow in the Environment and Natural Resources program at the Harvard Kennedy School. She was a national science reporter for the Washington Post, past president of the National Association of Science Writers and the Council for the Advancement of Science Writing. She was for many years a panelist on the public broadcasting service show Washington Week in Review. Her research focuses on science writing and how to improve news media coverage of controversial science policy issues. This lecture, supported by Chris, debuted in 2012. Its goal is to introduce Mills students to mentoring and advancement opportunities by bringing in top leaders in science to our campus. I'm grateful that Dr. Bargmann and Chris were able to spend the day actually on campus talking to some of our students. Please join me in welcoming Chris Russell to introduce our speaker.
Well, it looks really great from up here. I hadn't seen how the room filled in, and I think we are in for a treat. It's really my pleasure to have actually spent the day with Cori Bargmann on campus and meeting faculty and students, and a very impressive campus, and also very excited about Beth's participation in all of these Women in Science initiatives. So it's my pleasure to introduce Cori Bargmann. She is a distinguished neuroscientist, geneticist, professor at Rockefeller University in New York City, and more recently, she came back to San Francisco to be the head of science for the Chan Zuckerberg Initiative. So she still has a lab in New York, and she has her feet firmly planted on both coasts. So she managed to commute back and forth. Today's talk is Neuroscience, Technology, and Collaboration, a personal view. She's going to talk about her own work in brain research and the nervous system, as well as the frontiers of science that the Chan Zuckerberg Initiative, $3 billion worth, is tackling, which is really exciting. She is also the head of the Lulu and Anthony Wang Laboratory of Neural Circuits, okay, that's a long one, and Behavior, and is the Torsten N. Wiesel Professor at Rockefeller University. Her own research has been in the sense of smell, and she's really been a pioneer in connecting her own research on I can't say lowly worm, but a very famous worm, C. elegans, which provides a roadmap for understanding the brain and neural circuitry, I guess we would say. She has won countless awards and had honors, including the 2012 Kavli Prize in Neuroscience and the 2013 Breakthrough Prize in Life Sciences. She also co-chaired a collaborative effort with the National Institutes of Health Committee, setting goals and strategies for President Obama's Brain Initiative, which was a very important initiative. She is no stranger to the Bay Area, and so I'm a little jealous that I'm still stuck on the East Coast, and she had been at UC San Francisco for almost 15 years before going back to New York and Rockefeller University in 2004. She was introduced in 2016 as the head of science at Chan Zuckerberg by Priscilla Chan, a physician, and I guess maybe had been a fellow researcher she's a physician at UC San Francisco. And Priscilla Chan introduced Cori Bargmann as one of the world's most respected neuroscientists and geneticists, and a legitimate rock star. I will say in science, but I hope we have more rock stars. And in fact, that's part of what Mills is trying to achieve. And we have a great privilege of hearing this wide-ranging experience she has had as a leader in science. And again, I'm very proud of Mills for the faculty and students and the efforts in women in science and creating the next generation of leaders in science. And there's still a lot to do, but I think we're excited to have Cori Bargmann give us some of her insights from her bicoastal research. So thank you, Cori.
So I'd like to start by thanking Mills College for the great honor of being invited to be here. And I'd like to especially thank Chris Russell for being here. She has been involved in biology and some of the policy issues around it from the very earliest days of the molecular biology revolution. And it's an honor to spend time with her and with you while I'm here. Now, your motto here is there are different paths to one destination. And my talk today will be about my path to a destination for leadership in science and the work that I've been trying to do at different stages in my career and how I've learned from that how to think about perhaps ways that we can make science better. And my story starts in graduate school at MIT, where I was at the same time as Lisa Ori, and where I studied cancer. And I just want to talk about what I learned there. So I, as a graduate student, studied this very fine organism, which is a rat. And this particular kind of rat got a particular kind of cancer. It was a brain cancer, a neuroblastoma. And we were trying to understand the genetic basis of this neuroblastoma. And I was working in a lab where everyone else was studying human cancer, Bob Weinberg's lab. And there were many reasons that it seemed much more interesting to study human cancer, and it seemed like kind of a weird thing to be studying this rat. But the reason that this rat was interesting was that the cancer that arose in this rat was associated with something called a tumor antigen that could allow the immune system to reject that cancer and allow the animal to survive. And this tumor antigen we characterized in collaboration with someone who had very different expertise from us, a man named Mark Green, who was an immunologist. And ultimately, my little piece of the story is that I identified the gene, I cloned the gene that gave rise to this tumor in the rat, and determined that this gene actually encoded a protein that sat on the surface of the cancer cells. And that was why those cancers, so first of all, that was the tumor antigen, and that was why the antibodies could attack the cancer, because the cancer couldn't get rid of that gene without stopping being a cancer. And so it maintained that, and it's sort of the same thing that made the cancer grow was also its greatest vulnerability. Now, this was an interesting insight, but it seemed very rat-like, and there were lots of reasons for thinking it wasn't relevant to human neuroblastoma. But after my work was finished, and at that point it would have been stopped, other scientists picked up the threads. So Mark Green developed really good reagents that allowed you to detect this molecule, the tumor antigen. And then working together with a medical scientist at UCLA named Dennis Slayman, they looked at a lot of different human cancers, and they discovered that, sure enough, it's not altered in neuroblastoma, but it's altered in a subset of breast cancers. And in fact, at the time that that was discovered, that subset of breast cancers was the most lethal and most dangerous form of breast cancer for women to have. The gene involved, we called NU for neuroblastoma, but it's also called HER2. And after that discovery was made, so there was a relationship to human cancer, but it took this medical group and the immunologists to find it. And after that discovery was made, the story migrated to one of the brand new biotech companies that was growing up in the Bay Area called Genentech. And Genentech made an antibody against the human version that could be tolerated by humans. And that antibody is called Herceptin. And it's now used to treat about 100,000 women a year with breast cancer. And through that antibody, now that particular form of cancer is the kind you actually want to have because it is one of the most treatable and one of the most curable. So what I learned a set of things from this, right? And what I learned is that scientists build on each other's work, and that it takes a lot of different people with different expertise to make progress. And I do not want to exaggerate my contribution to this story. I'm one of hundreds of people, maybe thousands of people who contributed to this work. But I did learn from this that science works, and if you really try to deeply understand something, and if you work with other people with different expertise, you can make progress even against some of the most terrible diseases of people. And that is an inspiration for me, and I think an inspiration for all of us in science to realize that even when you're doing something that seems awfully obscure, and even when you're laboring away and the experiment hasn't worked 10 times, maybe that 11th time you'll do something that someone else can build on, someone else can build on, and that ultimately will lead to progress that makes a difference. So from there, we're going to move to the next stage of my career, and I'm going to tell you a little bit about some of the things I learned there from thinking about the brain. So I am a neuroscientist, and I just can't really imagine why anyone would not be a neuroscientist. So this is a problem. But I just want to point out that the human brain is what makes us human. It's the source of our emotions and our memories and our thoughts and our perceptions. And it's an absolute wonder of nature. And I want to know how it works. I want to know what happens and where and when and how. And there's a problem, though, which is that the human brain is really, really, really complicated. So the human brain has 86 billion neurons, which is more than there are stars in the Milky Way, right? It has 100 trillion connections between those neurons. Now, thinking about that makes it possible to understand how it can work and do the things that it does in principle, but how do you figure that out in practice? And so my own lab has been trying to address this using an approach that turns out to be really powerful in biology, that's based on something that's almost like a trick. And the trick is that the brain is ultimately assembled through the actions of the genes that are passed on from parent to child in every single human being. And those genes specify the development of the brain, its functions, its capabilities, and its properties. And those genes, remarkably enough, turn out to be shared by humans and other animals. So they are like Lego pieces or building blocks that can be used to assemble many, many different kinds of biological structures. They get put together in different combinations. And I have to say, the extent to which this is true came as a surprise even to those of us who studied animals, even to those of us who studied simple animals, which I do. So maybe it doesn't surprise you that humans share 99% of their genes with chimpanzees, although it's kind of surprising. But they also share, well over 90% of their genes with like rabbits. And even if you get down to things like butterflies and worms, somewhere between half and 3/4 of genes are recognizably similar between these different organisms. And so you can use this trick of studying genes in simple organisms to figure out what do they do, how do they work, how do they talk to each other, how do they give rise to biological properties, and then you can try to use that to understand something more complicated. So here is my metaphor for that. The human brain is a Boeing 747. It's a fabulous aircraft. This slide is working a little less well this week than usual, sorry. And it's an incredibly complex, fabulous machine. And maybe we can try and start by studying something simpler that doesn't have the same capabilities. This is my mother's 1968 Volkswagen Beetle. Let me tell you, when you open the hood of the Volkswagen Beetle and you look inside, you can look under the hood and figure out everything about how a car works just by watching. It's right there, things go around, you're like, oh, that connects to the wheels, it's really easy. And for us, looking at a simple animal is a way of looking under the hood of what a brain is and discovering what genes are doing and how those things work together to give rise to different functions. And here is the organism whose hood we look under. This is the tiny nematode worm, Caenorhabditis elegans. It's something called a synanthrope. It's an organism that lives in association with humans. It lives in our gardens and in our compost heaps, and it eats bacteria. And the reason we love it is that, first of all, it's transparent, so we really can look under the hood. We can look at this animal and see what's going on in its brain while it's thinking, which is pretty amazing. And second, it grows really fast. It has a 3-day generation time. We can generate animals that are genetically identical by the thousands and study them, and then change one gene, generate thousands of those and study them again. And so we have a tremendous ability to kind of make those relationships work. And furthermore, through a heroic effort made by John White and his colleagues in the 1970s and 1980s, we have a complete wiring diagram of this animal's brain. It has exactly 302 neurons, and those are all shown on this slide, not that you can read them or anything, but each one has its own name, and each one is exactly the same in every animal. And like if I go and point at a neuron, or if Jared goes and points at a neuron and says, this is ASH, then every single person knows they're looking at the same neuron, and they're talking about the same neuron, and it has the same connections in these wiring diagrams, and it expresses the same genes, and we can build on each other's work, and we can really try to understand the system as a whole. So we know that it has 6,900 what are called chemical synapses, connections between these neurons, and it has 890 what are called gap junctions, electrical connections between these neurons. And we have the whole wiring diagram in front of us, and we can try and figure out how that's working to give rise to the animal's behavior. And I'm just going to talk about a couple of its behaviors today, but I just want to mention in passing that with 300 neurons, this animal can do pretty much, actually. It moves around, and it mates, and it recognizes members of the same species, and and it forms memories about food and things that have been experienced in good environments and bad environments. It's, you know, it's pretty successful with a pretty limited brain material. So we can study the basic biology of this animal and get at some questions that we think are more interesting for more complex animals like ourselves. So here's an example of a question that I think is a really good question, and that is, how does the brain change under the influence of things like different motivational and emotional states? And there's an important insight that we've gained from that from studying humans and human psychology and human neurology. And that is that there's a class of molecules within the brain that are important in regulating motivational and emotional states. And those molecules are small molecules called neuromodulators. So neuromodulators are perhaps, they're not the molecules that mediate the super fast communication of nerve cells with one another. They're not the molecules that mediate rapid millisecond level signaling that would let you recognize your sister's face or hit a baseball. What they are slower acting molecules, and they're made deep within the brain in those little colored spots here, shown here. And they act at a distance across the brain, and you know, they're not so interesting to a neurophysiologist because they don't make neurons fire action potentials, and they're not so entertaining to a neuroanatomist because they aren't released at like the chemical spine synapses like the ones that are mapped out in C. elegans that I mentioned. But just to give you a sense of how important they are in humans, they are the targets of most psychiatric drugs and most drugs of abuse that are used by human beings today, and even some neurological drugs. So some of them are pretty famous molecules like dopamine, which is, you know, when dopamine systems become malfunctional, people develop Parkinson's disease, and it's also the target of drugs like Ritalin and cocaine. Serotonin, the target of the major classes of antidepressants that are used in humans. Norepinephrine-related arousal compounds. And also, in addition, about 100 different peptides, small peptide molecules that fall into a number of conserved families. That include, you know, importantly for our current society, the endogenous opioids, which are the targets of molecules like morphine and OxyContin and fentanyl. So these molecules clearly have profound effects on motivation and behavior in humans. And they're implicated in things like sleep and hunger, mood, arousal, social drive, pain, and stress. So what do we know about these? Well, what we know about them is that they tend to signal through biochemical pathways mediated by G-protein coupled receptors. There's 100 plus of these molecules. There's a couple hundred of these receptors. They're all over the brain. And like, here's my question. What are they doing? Where, what, how? How do they affect the function of the brain? And I'm going to focus on one of these molecules to start with. And that's a molecule that's pretty famous for a molecule. And that's a small molecule called oxytocin. It's a small peptide, and it's a neuropeptide first identified in sheep that is present in humans as well, and it regulates mammalian reproductive behavior. So the most famous feature of oxytocin, and the time many of us have been exposed to it, is that when a female mammal gives birth, oxytocin is massively released into her bloodstream. It causes the contractions that give rise to the birth of the baby. It causes the milk letdown response that allows the mother to feed the baby. And it also causes changes in the brain of the mother that modify her behavior. It's important in mother-pup bonding, and it's important in maternal aggression. And oxytocin and some closely related peptides also regulate things like social recognition and pair bonding. So many of the women in this room have probably been exposed to an oxytocin agonist called Pitocin, given during labor. And all of us, women and men, young and old, are exposed to this molecule being released in our brain at different times throughout life. Now, this function, particularly the functions in social recognition and bonding, have caused oxytocin to just invade popular culture like a bad weed. And so, for example, you can buy oxytocin candies. here on Valentine's Day to give to your loved ones. You can go on the web and get instant trust oxytocin spray. You can get clinically proven Dr. Oxytocin Factor to improve your love life. And there has even been a book written called The Moral Molecule, the Source of Love and Prosperity, about oxytocin. May I remind you that oxytocin was discovered in sheep? I don't think so, right? So clearly there's something here that's resonating with people, but it's gotten a little bit out of hand, and it's really worth figuring out what it does. And just to reinforce the notion that this is not a moral molecule, oxytocin turns out to also be present in the nematode worm Caenorhabditis elegans. And it is present in a recognizable form where you can tell that it's really derived from the same common ancestor, and it has the same kinds of receptors. And where it's present, it turns out, is in some structures in the tail of just one sex of the nematode worm, and that is the male sex. So in C. elegans, there's a male, which is the little skinny guy, and then there is a hermaphrodite, which is basically a female. And the male needs oxytocin to carry out its mating behaviors. If it doesn't have oxytocin, it gets kind of confused during mating. It doesn't know who its mate is. It meanders off. Everything gets kind of repeated and disorganized. And it does that through the release of oxytocin from a particular neuron, a neuron that we can see, a neuron in the tail of the worm. And it releases oxytocin onto a set of other neurons that we can see, whose receptors we can identify. And we can follow the activity of these neurons while the animal is mating and try to think about what they are doing and how. And so this little trace here shows you what that one neuron that makes oxytocin is doing while a male is mating. It's the recording from that neuron for about 5 minutes of a mating behavior. And at first the neuron is really quiet, and then the male gets with the program. He's like, I get it. And at exactly that point, the oxytocin neuron shoots up. That's where the green trace starts. And it does some pretty complicated dynamic things during these active stages of mating. And then he reaches a point where sperm is being transferred, and he's like, got it? And the oxytocin neuron turns off. And that's what's happening in the yellow part of this trace. So we can monitor this neuron and really show this neuron has a marker. It acts as a marker of a coherent behavioral state, mating behavior. And it's active during that period in a complex pattern. And its activity helps to distinguish an animal from knowing that it's mating from doing other things. And there are a set of things about this neuron that turn out to be kind of different from a lot of the neurons that are best characterized in different brains. Like most neurons will turn on and off in a time scale of milliseconds to do really fast signaling. And this neuron will be active for minutes at a time. And so when you start to think about things like a motivational state that turns on and turns off and might last for a longer period of time, you can actually relate that to what a nerve cell is doing and think, oh, these neurons have special properties. And it turns out you can identify them kind of reliably based on their gene expression patterns now from other systems. So this is an example of how you can study a molecule in one animal and think about what it's doing in other animals as well. You know, obviously the reproductive behavior of C. elegans is really, really simple compared to the reproductive behavior of a mouse, let alone a human, or the parental behavior. But you're still seeing this molecule tied into reproductive behavior, even in very simple animals. It's like having a word that means the same thing in different languages. Oxytocin is tied to this behavior, and it's tied to this behavior actually across vertebrates and invertebrates. It gets kind of locked in to modify this behavior, and then you build more and more complex assemblies onto this molecule. So that's one view of what a molecule does in a behavior. I'm now going to give you kind of an equal and opposite view, again, of thinking about how we can think about these sorts of complex behavioral properties in different animals. And first I want to remind you that mating is not the only social behavior. For example, there are a lot of us here in this room. And in fact, most organisms, increasingly we understand that most, perhaps all organisms, interact with other members of their species, that this enables them to be more successful in many different ways. And that could be butterflies migrating over thousands of miles, or it could be fish cooperating, or it could be herds of beasts, like in this case wildebeests, avoiding predators by herding, or in this case, it could be a group of worms, some of which are labeled here so you can see them more clearly. This is a cluster of about 30 animals. And we find that some of their life cycle, C. elegans chooses to spend in association with other animals. And after kind of a lot of understanding of that, we actually have some idea of what that behavior is for. It turns out that the animals do it some of the time and not other time. It's a stress-induced behavior to aggregate, which actually aggregation is also very commonly a stress-induced behavior in other animals, where it helps them cooperate. And like mating behavior, it turns out to be regulated by neuromodulators. In this case, really importantly, modulated by a neuropeptide receptor. So it's on the receiving side. I told you about oxytocin, which is on the sending side of a motivational state. This one's on the receiving side of a motivational state. And this is like the anti-oxytocin. This is a molecule whose job it is to suppress social behavior. When it is active, which it is, for example, in the British worms shown there to the left, the animals are kind of scattered all over the place, paying no real attention to each other. And when it is inactive, as shown by a Hawaiian strain shown there to the right, the animals gather into these large feeding groups, into these large aggregates. So we can discuss the different traits of Brits. We can make some Brexit jokes now. Be that as it may, what I want to just really talk about is the fact that having a molecule and having this animal where you can look under the hood actually lets you figure some stuff out. So, aggregation. Why do animals aggregate? Well, they aggregate because they make pheromones, small molecule chemicals that sort of mark them as members of the same species, and the other worms smell those pheromones, and that enables them to gather together. And from a set of studies in the lab, we were able to identify the neurons that sense pheromones, and those are neurons in this wiring diagram there, circled in red about in the middle. And then we have neurons that make this neuropeptide receptor and modify the behavior. And that molecule, those neurons are circled in kind of this area over here. And so this is not super helpful. But now that we have these 2 points, here's how you smell the pheromones. And here's something about whether you respond to them or you don't. You can dive into that wiring diagram and look at it in more detail. And what you see is something actually pretty cool. And it explains something that we had a lot of trouble with for a long time, which is, how come if we have a wiring diagram of the whole worm, how come we can't read that wiring diagram? How come I can't look at it and just tell you, oh, when worms smell pheromones, they're going to do this or that? And the answer is that the wiring diagram is ambiguous. It includes connections that can mediate both attraction from pheromones and repulsion from pheromones. They're both sitting there all the time. And so when animals are in an aggregating prosocial mode, there's a set of connections, it's actually a gap junction, to the neuron that makes the oxytocin receptor, and that mediates a strong attraction. But hidden in the background, not being paid attention to, are a different set of neurons connected through a different set of connections that actually mediate repulsion. So in an animal in which the neuropeptide receptor is not active, the pheromones are produced, the animal detects them, and the animal aggregates. But in an animal in which the neuropeptide receptor is active or is activated, the attractive circuit turns off. And now the repulsive circuit is unmasked. And so the animal now selects a different set of connections, and it reroutes its information flow. So the neuropeptide is switching the animal between 2 different ways of responding to a single smell. And just to maybe refer to something that you might have experienced in your own life that would be an experience of that, if you're hungry and you smell an attractive food odor like barbecue on the grill or something, you'll be very interested in it, you'll be oriented toward it, you'll pay attention to it. It's a very motivating stimulus. If you're not hungry, same exact smell is, it's aesthetically pleasing, but it's just not as motivated. And if, like me, recently you have a bout of infection with norovirus, when you smell that smell, you are immediately going to feel intensely nauseous, right? And that is the exact same smell, but it's basically 3 different motivational states that are routing that smell to different parts of your brain. It's not forcing you to do anything, but it's giving you some sort of fast information about what might be the right response in this circumstance versus that circumstance. And now taking a step back from that, I just want to return to that wiring diagram that we couldn't read. And I'm going to show you, like, here's part of that wiring diagram, including part of what I showed you. And it's incredibly strongly connected. It's not like one neuron attaches to another neuron. The neurons are like in this big giant tangle, and there are lots of different connections. And so what we think is that actually only a subset of those connections are really available at a given time. And that's what the modulator is doing. It's sort of rapidly tuning some of them up and tuning them off so that information flow can change. And I just want to now return back again to thinking about more complex brains and tell you that the human brain is also really, really, really overconnected. It's incredible how many different connections there are between how many different brain regions. I mentioned those 100 trillion connections between neurons, and that seems really daunting. But in the human brain, there are these modulatory systems, and the suggestion would be, and a testable hypothesis would be, that these modulatory systems are basically making it so that some of these connections are more or less available at different times, and that this might give us some insights into how the brain becomes not just one brain, not just a passive brain, but sort of a fast, rapid, adjustable, and flexible brain. So to summarize that part of this part of the talk, the part about what my lab works on, we are led by this because we can look at the same brain under different circumstances with different genes present or active or inactive. We can understand something about a complex biological process, and we can make a statement about what we think is going on. We can say, you know, there's a wiring diagram, and that was the whole reason I went to work on the worm, but it's just a set of options. And the neuromodulators help select among the options to modify behaviors. And then it gives us a whole set of things that we can look for in these neuromodulators in other systems. So for example, it turns out that they have a completely different logic from the fast circuits at the level of anatomy, as well as at the level of physiology and timing. So you have to look at them in a separate way anatomically. We think they select among the anatomical connections to reroute information flow. We think that they organize higher order coordinated circuit states like mating behavior or aggregation behavior, not the individual sort of fast actions, but sort of the overall knitting together of a state. And finally, from this and other work, we think that they operate over long time scales, which could be minutes or hours or even days to modify the behavior, but then can be rapidly reversed to return a circuit to its previous state. So these are discoveries, they're ideas, they're in some ways developed from looking at things very narrowly, but once you have ideas like this, you can move out and try to think about what other principles they might be applied to and what other situations they might be at. So at this point in a talk, it's traditional for the speaker to thank all the people who were involved in doing the work. This is actually a picture of the members of my laboratory at the Rockefeller University. And it's also traditional to list some of the people who did the funding, who provided the funding for this work. And so here are several different organizations that funded the work that I talked about here. But you know, when I think about the work that we did, I've realized that there were people who are not acknowledged, who are as important to this work being done as the people who did the individual parts of it. And those were people who provided resources and technology that made those experiments possible. So let's just start with the wiring diagram. Everything we do is based on the wiring diagram, which was published in 1986. I have a first edition. It's this tattered, ripped up, pages falling out of it thing that I still look at like once a week to figure out what those connections might be and how I should think about them. And then there's the genetics. I just kind of glossed past that really fast. Oh, this gene that, this other gene that, this expressed there. Well, that was based on an incredible amount of work that was done by the people working on the Genome Project. And they made it possible to move back and forth between saying this gene, and now it's manipulated, and now I moved it to this other location, and I saw this outcome. And that's something that people don't always acknowledge when they talk about their work. But it made everything 10 times faster. And then there was, a lot of engineering of different sorts. So there was like physics engineering, like a particular laser that was really important in our work. And then there was microfluidics, a technique that was moved from the software industry back into biology. It was the silicon chip wafers got moved back into biology to help us work with these microscopic organisms. And then there was biological engineering that made these very cool fluorescent proteins that would light up when a neuron was active. So that's when I said I could see the worm's brain while it was thinking. I can see that this that this molecule lights up and tells me this neuron is now active. And then there's optogenetics, which I didn't mention, which lets us beam thoughts into the worm's brain by turning neurons on and off. And all of these things just kind of happen in the background, and then it's like, oh, blah, blah, this experiment. But you look at all the people who did this, and sometimes it took them decades to get this work done, and how important their work is, not just for us, but for everyone in the field. And then there's a lot of other people here in the background, long-term mentors and scientists that my lab published numerous papers with, who taught us things we didn't know anything about, like computational neuroscience, Larry Abbott at Columbia, or chemical biology, Michael Marletta from Berkeley, or population genetics, Leonid Krugliak from, who's now at UCLA. And all of these people are, again, it's easy to sort of feel like, well, I'm a really successful scientist, and then be like, but really, what we have is a really successful endeavor where a lot of people are working together. And so just sort of thinking about that provides a different perspective on work than thinking just about your own lab. And there are other sort of periods of time in which other perspectives have sort of become apparent. As Chris mentioned, I spent a fair amount of time working with the NIH on planning the BRAIN initiative. At other times, I've been on the board of a drug company and thinking about how drugs get developed, or on advisory boards for foundations. And all of those things, they were all really interesting and compelling in their own right, but they also all serve as paths toward a destination. So they're diverse paths that led to a destination, in the sense that somehow when these people here contacted me, Mark Zuckerberg and Priscilla Chan, and they said, we want to do something important in biomedical science. What do you think it would take to do that? I felt that all of these experiences I had had in cancer, in neuroscience, in technology, in working with government funding agencies, working really with pharmaceutical and biotech systems, had somehow prepared me to have an answer to those questions. And maybe I think some of the time is you don't even know what the path is that you're on, and then you find yourself there. So these two people, actually 4 people are in this picture, but the 2 adult persons in this picture are Mark Zuckerberg and Priscilla Chan. Mark Zuckerberg is well known as the founder of Facebook and a software engineer. Priscilla Chan, his wife, is a child of immigrants, actually of refugees. She was an educator. She did elementary school education, and then she went to medical school. She is a practicing pediatrician. And on the birth of their first and oldest child, Max, about 3 years ago, Mark and Priscilla decided that they had been very fortunate in their lives, and that their good fortune had allowed them to have this great success. But they wanted to make it the world such that maybe you didn't have to have good fortune to be so successful, and maybe to give more people more of a chance to be successful without having good luck. And so they founded the Chan Zuckerberg Initiative, to which they will give away the majority of their wealth in their lifetime, with 3 areas in which they feel that they can try to make a difference to make the future more open for everyone. And they're really areas that are based on Priscilla's individual experience. Education, because education is an opportunity for all. Biomedical science, because as a pediatrician, she really saw how important science was for moving medicine forward. And health is a great equalizer. And finally, justice and opportunity, again, as a child of immigrants, and as someone who grew up in a not very fancy environment, seeing how things like criminal justice system and immigration have huge influences on people. So they have set up an organization with sort of 3 equal pillars and 3 equal groups. And they asked me to be involved to lead and to set up the Biomedical Science Initiative, which, as Chris mentioned, they are donating $3 billion to over the next 10 years. And when they talked to me about this, I said, well, what is it that you want the initiative to do? What is your hope for this? And they said, well, we want our initiative to support basic science and technology that will make it possible to cure, prevent, or manage all diseases by the end of the century. And I thought about that for a while, and I want to say that when you first think about it, it sounds arrogant, and then when you think about it more, it's an act of respect for how far medicine has come. We're talking about 80 years. Let's think about where medicine was 80 years ago. the very first antibiotics were being developed, right? 100 years ago, 3 of the top 10 killers of human beings were bacterial diseases, which we now have antibiotics that we can just cure. That comes out of science. 100 years ago, we did not know that cigarette smoking caused cancer, but now we can prevent cancer if we don't smoke. We did not know that high blood pressure and high cholesterol led to heart disease and strokes. And so there too, we can prevent those diseases now. Things like the statins and the high blood pressure medications have made these diseases much, much more serious, even since the 1960s and the 1970s. And in cancer, my field, there's been a kind of sustained effort after a very long slog, where for the past 25 years, death rates from cancer have been going down 1% a year in a pretty disciplined manner because of the science that has enabled cancer medicine to move forward, most recently by great advances in immuno-oncology. So if you look at that and you see how much progress there's been, HIV going from being a fatal disease when it really a 6-month death sentence when it was first discovered in the 1980s, to now being a disease where if you're diagnosed at age 20 and you have access to a great healthcare system, your life expectancy is 70. These are really great success stories, and we should celebrate the fact that science works. And I started by telling you that science works, okay? So I believe this firmly. But I also believe that 81 years, it's probably not enough if it happens at the current pace. So it has to happen faster. And so I said, you know what we really want to do is we want to accelerate biomedical science. And I have ideas about what would make that happen. And one is that tools and technologies are a good way to support science. And that the more you can get different people with different backgrounds to work together to solve hard problems and collaborate, the more that you'll be able to accelerate science as well. So our 10-year plan is to see if we can accelerate biomedical science. And the good thing about this goal is that it doesn't say we're going to cure all diseases. It doesn't say we're going to do the work ourselves. It says we're going to enable the fantastic community of scientists and interested people out there to do this work by making their work faster. And I have to say, this is also something that, you know, the engineering perspective really likes that, because an engineer thinks he can make anything 10 times faster. So we want to do 1,000 years of progress in 80 years. That's our goal. And somehow that doesn't seem so bad. So this is our mission. It's to accelerate biomedical research. And here are our 3 pillars. The first is collaboration. And that is, we want scientists to work together, we want scientists to work with physicians, we want patients to be part of the equation, and we want engineering as well to just try and make the whole system more collaborative, and we want to support that. Second thing is enabling tools and technologies. And our symbol for that is the microscope, because before the microscope, nobody knows that the human body is made of cells. And after the microscope, everyone can see that for themselves. Everyone is a better scientist with better tools in their hands. And so it's enabling a whole field. And then the third pillar is building support for science. And I think Chris would like this. We have a separate policy arm that I won't be talking about very much, that's trying to think about how to build public support for science, how to give increased funding for science, and both from public sources like federal and state sources, but also from private sources and philanthropies, but also really to open the engagement into science from a broader level. I'm actually kind of proud to say that we are supporting the University of California, Santa Cruz's science writing program as part of that effort. So our beliefs are we want to support great people, technology, collaboration, and we're also we want people to share and build on each other's work faster, so we're supporting open science. How to do that? So here's our approach. We fund grants and partners on different scientific projects. We have in-house a group of software engineers and who are actually building different kinds of computational tools and software as one of the kinds of tools that can advance science. And we collaborate. We have a group of computational biologists who go out in the field, learn on the ground, collaborate with scientists, including our grants and partners, but others as well, and bring back the problems to the engineers to solve. Because it turns out you need a translation layer between an experimental biologist and a software engineer who really understands the advantages and the abilities of each side so that you can figure out where the added value is. And the collaborators do that. So just to kind of iterate how we're doing that, one of the easiest ways to communicate that is to say who we are. So if you went to our offices in Redwood City, who would you find? And you would find a group of people. So first of all, at the top, you would find a group of science program people. So these are, for the most part, people who are trained in science, and some of them are really heavily involved in grant making and trying to select people and programs for grant making. And then in addition, we have the computational biologists who go out and do work in the field. We have a communications group, so Leah is here from that group now, that's thinking not just about how do we communicate about Chan Zuckerberg, but how is science communicated more broadly. And then we have a policy group led by Tanya Simoncelli, who's really deeply engaged at the moment in thinking about bringing patients as more active participants in the research endeavor, not as subjects, but as true partners in research. And then at the bottom, we have a bunch of people that you might be surprised to see at a traditional philanthropy. And these are software engineers and product people and user experience people, people from Silicon Valley, people who've often been trained at places like Google or Facebook, and who have decided to come back and work in a nonprofit domain, because there's been tremendous progress in these areas in the for-profit world, and many of these ideas have not come back into science and education and policy. And so we're bringing some of them back. We're bringing some of them who are motivated to join us to work on these kinds of problems. And it turns out it takes a lot of people to build software. So actually that wasn't all of our software engineers. Here's the next page of people on the software system. It takes a pretty large group, and figuring out how to deploy this group of people in the right way is one of our biggest questions. And figuring out how they're going to work with the people we fund is also really important to us. So how do we choose projects? We're supposed to be solving all diseases, right? Where do you start? Do you make a list of 11,000 diseases and check them off one at a time? Do you try and pick big things that people are working on? Do you count the number of deaths per disease? You got to have a theory, and you got to have some advice. And so we are really thrilled to have an incredibly impressive scientific advisory board of people who are both great scientists and have done really innovative things in leadership in their own careers, who spend a lot of time with us, who come to our meetings, help us plan things, come to quarterly meetings to discuss what they think the opportunities are, and are guiding us in our work. And just to mention, Harold Varmus is a Nobel laureate and the former head of the National Institutes of Health. Bob Tejan from Berkeley is the former head of the Howard Hughes Medical Institute. Art Levinson was the CEO of Genentech and is now the CEO of Calico at Google. They're really a set of just incredibly impressive people here who bring their wisdom and their leadership knowledge to us to help us decide what to do. And then, but this is just, this is one group of people. We think that really to make a real impact, we can't just look at the senior people for what's been tried. We have to go out and talk to the people on the ground and ask them, if we want to accelerate science, what's slowing them down? And so we spend a lot of our time with scientists. We host meetings, we go to workshops, we have hackathons, we go to other people's meetings, we go visit people at their site, and we ask them over and over again, what's slowing you down? Where is it that you're spending your time that you wish you could make things faster? And we think about that in the following terms. We don't want to support one group or one institution or one project. We want to think of ways in which we can have a systemic effect, where we can pick a whole class of problems and do something to speed this up. And second, we want to do something where we're not doing what everyone else is doing. We want to find the places where there are gaps and where there are holes and fill in those gaps rather than reinventing the wheel. And as generous as Mark and Priscilla are, our budget at CZI for science is 1% of the budget of the NIH, right? So we are not going to make a difference if we just throw a couple more pennies into the big pot. We have to think about our own point of view and our own attitude. And so I will tell you about a few of the projects we've started with that in mind, to give you a sense of how we're thinking of this idea of people and technology and collaboration as a way of getting people to work together. And one of those ideas is right here in the Bay Area. It's a new kind of collaborative research site called the Chan Zuckerberg BioHub. And it is our effort to get the scientists from the Bay Area schools to work together in ways that they have not worked together in the past. And so there is space in San Francisco. That space is led, the leaders of this research site are two professors, Steve Quake from bioengineering and applied physics at Stanford, and Joe Derisi from biochemistry and biophysics at UCSF, are the co-presidents of the BioHub. They maintain their affiliations at the university, and they are building a group that's focused on technology development and advanced technologies, in which they are linking together many scientists and engineers in the Bay Area to work on new methods, particularly in infectious disease, Joe's area, and single-cell technology, Steve's, but with a broader idea. So there are about 100 faculty members now at UCSF, Stanford, and Berkeley who have joint appointments at the BioHub. They meet there on a regular basis to collaborate with each other and collaborate with some of the in-house staff. And they are starting to try to build up new relationships in this kind of relationship where they're not removed from their home institution, but they have another place where they can collaborate together on technological problems. Now, in addition to these scientists, the Bay Area schools, we've also embedded a set of our engineers into the BioHub to think about whether the right model is to put some of these engineers in a lab site and let them view the opportunities there. And through that work, actually, we have developed a software product, a platform, where the original idea was worked out by Joe Derisi. And it's a method that enables you to use a blood sample, or actually a urine sample, or who knows, any biological sample you want, and identify infectious agents, pathogens in that sample from metagenomic sequencing, even if it's a pathogen that's never been seen before. So the methods for this were developed by Joe in computational biology, and they're pretty cool, but the version of it that he developed cost about $2,000 per sample. And our software engineers kind of got their paws on it, and they ripped it apart, and they put it back together again. And when they were done, it cost about $18 a sample. And we are now partnering with the Bill and Melinda Gates Foundation to bring this to some of their field sites around the world, actually in 10 different countries, to be able to identify new infectious diseases in a global health context. And our first pilot experiment with the Bill and Melinda Gates lab led to the discovery of a new variant of chikungunya virus in Bangladesh that was causing an outbreak of pediatric meningitis that had not been previously seen. So it's collaboration on a larger scale, right? It's collaboration with the BioHub, with Joe Derisi, with the engineers, and with the Gates Foundation, and with their amazing record in global health and their ground game there, that kind of knits together these different groups to start to look at a class of diseases, infectious diseases, in a new way. What else? We're thinking a lot about tools and technologies. And again, we're thinking about this fairly broadly. I'm going to start with an example that involves software. And that is that we would like to accelerate the rate at which people can share knowledge and discover it, share it with each other and discover it. And so we are taking a mixed approach to try to advance the ability of people to share knowledge. We are supporting bioRxiv, which is part of the preprint movement that enables scientists to share their results with each other prior to formal publication. And we are building software tools that enable people to identify papers that are relevant to themselves in real time in a way that's personalized and predictive. It's like, this is going to be an important paper in your field in 2 years. Maybe you want to look at it now. So that's an artificial intelligence machine learning approach that's happening with our software engineers inside Redwood City. So that's another kind of tool. Again, that's a software tool. But like we have a very broad view of what a tool is. And here's another big project we're working on that we think is a tool for biomedicine. And that is something called the Human Cell Atlas Project, which is a desire to identify all of the cell types in the human body. So there are about 30 trillion cells in the human body. Nobody really knows how many different types of cells there are. Like maybe there's 10,000 different kinds, and maybe there's like 1 million different kinds. And it's not really known how many of each there are, and where they're found, and where their neighbors are. And there have been incredible advances in molecular biology techniques just in the past 3 or 4 years that make it possible now to think about solving this problem, really identifying what the cells are, what their molecular composition is, how many of each there are, and kind of use this as a foundation for learning about both healthy states and disease states. And this is a scientist-led project. It's totally grassroots. It's led by two brilliant computational biologists who are women, Aviv Regev at MIT and Sarah Teichmann at the Wellcome Sanger in the UK. There's lots of funders involved. And there's just a lot of excitement about figuring this out. And this to us is a perfect example of something that could affect all diseases, because every disease has a cellular basis. Every disease, there's some cell in your body that's either not doing what it's supposed to do or it's doing something it's not supposed to do. And if we know what all the cells are and we know what their normal properties are, that's a foundation for studying every disease in more detail, figuring out what's going wrong. So this is an example of something where thinking across diseases leads us somewhere that you might not be otherwise. And again, our approach is a hybrid. We're building the data coordination platform that's going to store all this data and make it available freely to all scientists around the world. And we're also funding the people with wet labs who are doing the work. And at the moment, that's about 200 different research groups in 15 countries who have just applied for us in completely open calls and have risen on their merits to work on this collaborative project. So there's more. We have a couple of other projects that we've launched just this year that we're starting to work on. One is to start to work again on a class of diseases, in this case neurodegenerative diseases like Alzheimer's disease and Parkinson's disease, where we're again trying to find ways of getting people to work together, explicitly bringing together doctors, scientists, and computational biologists and software engineers on a project. We're currently funding, I think, 48 groups in this area. We're funding other kinds of technology, in this case, both hardware and software for microscopy, because we think that imaging and microscopy is a really important area. And we're starting to support the staff scientists at core facilities who do that work that we think are really key people for education and innovation and dissemination of technology. And finally, we think that there need to be more kinds of different scientists. And just yesterday, we announced a partnership between the University of Maryland, Baltimore County, and the University of California, Berkeley, and the University of California at San Diego to bring more underrepresented minorities into science and technology education at the undergraduate level. UMBC has an astonishing record of supporting minority success in science, and we would like to see if that could be cloned in this state as well. So we think about things, shared tools, standards, formats, common languages, building on each other's work, and we're just trying these different ideas to see which ones might work. And what we have to ask ourselves is what does success look like? And that's what we really care about. We care about productivity, about the sort of traditional scientific success of this or that paper, but not just about that. We care about other kinds of success as well. We care about the reach. If we want to make a new kind of science, we have to reward people for being a different kind of scientist. We have to fund people not because they had the fanciest paper, but because they made the best code, or they generated a data set that everybody else could use, or they generated a protocol that suddenly moved a whole field forward. And so we're thinking about how to evaluate that and add that to our process, and the reach, the sharing of those things, and the ability of people to collaborate. So it's a little bit of a different set of values that we're trying to implant into our funding system. And then, we're just thinking about all the different ways of getting people together. That can be a communication tool like Slack or GitHub. It can be a meeting. It can be a hackathon. This is a real-life picture from one of our meetings with laptops out, including our computational biologists and engineers, but also grantees and some of our partners at BioHub working together, writing code and solving problems together. So we're looking for people who share our goals and our values. We want people to contact us. We want to broaden our reach and find ways of accelerating science. These two websites will tell you jobs for people that we're looking at, and also just let you know about the projects and tools and requests for applications we have open. I think that this is a good time to make this kind of a move in science. I think it's time for us to move beyond rewarding individuals for their individual accomplishment and really try to pull together for the big problems. And I feel incredibly privileged to be in a position to do that in my own work, and I'm trying to live up to the occasion. And finally, sometimes people say it's the destination that's important, and sometimes people say it's the journey. And I would say it's, but also really important are the crossroads where you meet with other people who share your values, where you trade your ideas, where you find out the next steps, and where you move on to the next steps of your different paths together. And I feel that here at Mills, I'm sort of at a crossroads that's uniquely valuable to me, and I'm grateful to be here. Thank you.
Dr. Bargmann has agreed to take some questions.
One thing that really resonated with me was the way you said that we need to collaborate more and we need to share more and we need to make sure that everybody has access to the information. Is there any platform where there's access to the experiments that failed?
It's really important to know what hasn't worked, because discarding the wrong answer is a step toward the right answer. And sometimes there's something that seems like the right answer, and everyone tries it over and over and over again, and then you don't make progress. And so you need to be able to share what didn't work as well as what did. And there are a few places in publication where that's starting to be explored. The Wellcome Trust in the UK, the Gates Foundation, and something called the Faculty of 1,000 are sort of starting to have places where you can publish well-done results that gave a negative conclusion or that didn't work. And I think that will be, kind of getting over your pride about I only have to be successful and publish my big successes is part of a process that will move science forward. So we don't have any great ideas yet, but we're watching.
What is slowing science down? Everybody trying, or a lot of people trying the same thing, and it's redundant and it's not working.
Exactly. That slows science down, and it keeps people from discarding that idea and moving on. Yeah. And most people don't want to do it because they're afraid they just mess something up, right? So it's like, I don't want to tell you that this experiment didn't work for me because then you'll just think I'm an idiot. But that's not the case. And so a well-done result that is something that you didn't expect or that didn't do what you expected is an important thing to share.
One of the questions I had was regarding your point, which is very true and is something we talk about at Mills, which is the importance of diversity in bringing science forward and bringing thinking forward. And I was wondering, what at your organization, what are the strategies that you do in order to recruit scientists from a bunch, and engineers and thinkers from a bunch of different backgrounds? And I was looking at the slides and there is some diversity, but I'm wondering what you guys do in particular to find those types of people.
So thank you for asking. I would just like to say that I'm really thrilled that the first two questions come from students, because usually students just sit there and the faculty ask all the questions. So thumbs up. Thumbs up. Okay, that's the first thing I want to say. And then the second thing is I'm really impressed with the seriousness that Priscilla Chan has taken to the idea of diversity in our organization and in the work that we do. And so we have, it's really hard once you have a non-diverse large organization to then go and fix it. And so really from the beginning, it's been an emphasis of the organization to try to bring in people from different backgrounds, to try to, as much as possible, represent the communities that we're working in. So we have a very strong technology component in the organization, and then in addition we have science, education, criminal policy, justice and opportunity. I think we're over 50% women, despite being a largely technology organization. I think that's been a really serious thing. Our engineering groups have a high fraction of women. I think we're doing really well there. And I think at this point we're over 20% underrepresented minority organization-wide. Science, I think, is 20%. We're kind of bringing up the rear, but we're working on it. So I think that it is something that we're just taking seriously from the get-go. And I think it's important because I think that we will do more important work by having more people in the room. I think that the kinds of, for example, in health, issues around disparities are going to influence what people work on. The things that you know from your own community are going to influence what you think are important problems to solve. And as much as I really admire and respect the National Institutes of Health after having spent a lot of time there, I also know that Congress funds diseases of certain cohorts more than it funds others. It does not fund child health with anything like the resources that it funds cancer and heart disease and diseases of the elderly, right? And so that, I think there are going to be lots of other things like that where you realize that people are falling between the cracks, and someone with, you'll be asking better questions and making better decisions with more people at the table. So, still, can't feel smug, can't let up, work to do, but we share your goals.
So, one of your big ideas is fostering collaboration, trying to keep people together and involved in a common endeavor. What kind of resistance have you encountered in Silicon Valley, where there's a lot of emphasis on secrecy and trying to monetize individual ideas for particular companies?
We have the privilege of being in a nonprofit setting where we are not trying to monetize, we are explicitly not monetizing the work that we do, because we are funded by a generous gift from philanthropy, we do not have to raise money, we do not have to sell anybody's data, we do not have to convince anyone that this is going to be the highest return on their investment in a financial way. I think that there is, among other things, one of the things that we've found helpful is that we're placing a big emphasis, for example, on the software side, on doing everything open source. Within the software community, there are closed source and open source mechanisms, and to the extent that we can make code and software freely available, that sort of takes some of those things right off the table. It's like either you're doing it or you're not, and that will help us do that. I don't think we're going to change the whole world, but I think biomedical research is a money-losing endeavor, and we're lucky in the US and in the world, frankly, that the taxpayer has been willing to fund it generously, and I think philanthropy is also on board for recognizing that this is not going to be the place that you make money in the short term, but it could be a place that you save a lot of money in the long term. And I should say, just an aside point, we're completely separate from Facebook. There's like no, but it's an important thing relevant to your point, because you're kind of like, so yeah, I hear you. and no, but from the point of view of Facebook, it was just a much cleaner thing to have the philanthropy completely separate. We have separate organization. We don't share information across these boundaries, and that keeps that cleaner. It makes it, just takes that issue off the table for us, which is good. Probably for both sides.
Hi, my name is Taja Lester. I'm an alum here at Mills who went on to work in biotech. I'm most recently from Genentech, so great to see you. My question is similar, actually. I'm wondering what's your typical sort of timeline for the projects that you're looking for? Are you looking for things that are going to be doing work 45 of the years out or 10 years of the years out? And I actually am wondering, is there any kind of path to commercialization, and how close are you to Genentech? I love seeing art, so thank you.
So the great thing about this end of the century time frame is the idea that we can really think about what we could do that would take 20 years to make progress, or even 50 years to make progress. And that's not a time frame that people always have in mind when they're making decisions about science. And in particular, it's not a time frame that people in the commercial sector are in a position to take, right? You have to, the next shareholders report or whatever is going to be the time frame in which you get your next judgment. And so we have the luxury of doing things on a longer time scale. That doesn't mean that we don't want to do things that could happen faster. We are in the business of trying to accelerate things. It just means that we can evaluate, and we think at this point, we think the gaps are maybe in the slightly longer term thinking compared to the short term thinking. And like you, I have a lot of admiration for Genentech and for the biotech companies and what they're able to do. You know, there's been a lot of really great medical progress out there in the past decade or two that comes out of these areas. So we don't want to duplicate what they're doing. We want to find the gaps. It's not to say that, but there may be gaps that end up in that place. For now, we've decided, you know, we're not going to do clinical trials. They're super expensive. We're not going to do drug development. It seems like chemical companies are good at that. We're going to do the earlier stages, and then we're going to hand them off to Art Levinson. And he's going to do the next stages of what that turns out to be. But we know that even for technology, sometimes the real path is going to be a path to commercialization. We won't do that ourselves, but we are open to others doing that. So for example, for all of the research we fund, we are not making any claims on the intellectual property. That intellectual property belongs to you, because we know that sometimes the best way to move things forward is going to be to have a company that actually then does that super expensive clinical trial. But we do ask that for any work that we fund, the intellectual property will be made freely available for all non-commercial purposes, including pre-commercial use by companies. So as long as it's accelerating science, that's our agenda. We don't want to make the money, you know, but if the right path forward is commercialization, we will not prevent that from happening. Whether we should do more in that direction over time, we'll see. Again, we kind of poked around a little bit in venture, just to sort of ask, like, should we be there? And the message we got is that that's not where funding is needed right now. There's a lot of really good funding in venture. The funding for the fundamental sciences is where the gaps are. So we're learning, you know. 3 years from now, I might give you a completely different answer, but we're trying to be smart, we're trying to learn, and we're trying to listen a lot. Yeah.
That's a good description of us at Mills, trying to be smart, trying to learn, and trying to listen. So please join me in thanking our 2019 Russell Women in Science Lecturer, Dr. Cori Bargmann.